Hello, everybody. So we're going to continue chapter 4, which is over cell injury and death, um, in 4 kind of smaller lectures instead of one big super long one. The first one is just going to be a general introduction to cell injury. So there's a few things we want to talk about up front just to get oriented. The first thing that we're going to look at is the fact that this is a unique cellular phenomenon, right? So we're not looking entirely at the whole organism just yet. We're not looking at how this moves through populations. We're just going to keep an eye on how things work at the level of the cell and at the tissue level, right? So cytology, histology. So generally we categorize cell injury in three general ways. And they each build on top of each other. The first type is a mild or short-lived change. There's some adaptation here. Cells are able to make a few modifications and just get through the stress, wherever its source is, and we'll look at a couple of sources later on in these subsequent lectures. But generally, this is reversible change. When a cell, however, is being exposed to what we consider a persistent source of stress, that adaptation becomes a little bit more inflexible, and the self becomes a little more committed to this adaptation style in order to survive or even maintain this tissue long term in the organism. So we're going to look at that. That's often it is sublethal, which means the cells are able to continue to survive, but persistent means the stress is sustained. Finally, we'll look at some examples of severe or prolonged cell death. So the severe prolonged cell death ends in a couple different categories. Generally, either the cell will die because of outside forces, or we'll look at, you might remember, apoptosis. Apoptosis was what cells use in terms of a programmed death. And so they would use internal mechanisms to program a cell lysis or disruption, terminating that particular cell, so that either the immune system could intervene at that point, or reversible cell injury is the mildest form of cell injury, and there are 3 categories. So reversible cell injury can be observed by a few phenotypic deviations from normal, let's say. Some of these include the accumulation of water, or actually pushing out a lot of water into the extracellular matrix. Sometimes there is accumulation of specific substances. We have some lovely photos coming up. We'll show you that. Others include lack of normal metabolic functions, and sometimes that comes from a lack of metabolites, right, or including nutrients from the outside environment of the cell. Or it can be the result of internal regulatory elements, such as perhaps gene silencing or decrease in enzymatic activity that can change the otherwise what we call here normal metabolic functions of these cells. The important thing to remember is when you're at this first stage is you can undergo stress, and again the cell changes a little bit to accommodate the stress, whatever it is, if it's lack of water, lack of certain nutrients, whatnot, but it's able to return back to normal after the stress has been alleviated and then there's been a proper amount of time to kind of heal up. One of an example of reversible cell injury can be seen here. This is hydropic swelling. So hydro, think, well, it's specifically referring to the hydrogens in water, right? So H2O. And dropic refers to a kind of, it's the seemingly old-timey, but it still happens, condition where individuals who experience severe edema or swelling, usually in response to some type of parasite or bacterial infection. Kind of got really swollen, often very distended bellies, and that's known as the dropsy. So hydropics, so this would mean this is a water-based kind of swelling of the cells themselves, and I'll show you a picture of that in a bit. Generally, this is a result of the inability to maintain balance across the membrane of specific ions, namely the sodium and potassium exchange from the sodium-potassium pumps. Also, lack of ATP, right, that cellular currency, can also lead to a disequilibrium of water balance in the cell. When you look at it under a microscope, what you generally see in cells are because they've taken on so much water, their cytoplasm actually looks pretty pale, right? And it's really just because it's super diluted. And it says it's large, but that, it's not just that it's a large cytoplasm, it's just that the cell itself is kind of engorged and distended. Other components include the endoplasmic reticulum is dilated and actually looks almost like a corona instead of a labyrinth around the nucleus. And finally, the mitochondria within, we remember that these are not just organelles, they are endosymbionts, right? So they have their own DNA and whatnot. So they also become very kind of swollen and distended themselves. Just to kind of give you a heads up, when referring to this type of hydropic swelling at the level of the organ, we often follow it with the suffix of megaly, right? So if I were to say splenomegaly, you would think a swollen spleen, right? So stuff like that. So let's go ahead and take a look at a picture of hydropic swelling at the at the cellular level. Here we go. So as you can see, this would be the outline of a cell right here, okay? And see how the inside of it looks very kind of that pale that we were talking about, right? That's actually distended cell, so it's actually just really swollen with water. And then if we look, let's see here, around, you can see this is the nucleus right here. Oops. Sorry, you can see the nucleus is right here in the middle. If I can get my highlighter to go. There we go, right here in the middle is an example of a nucleus. And then around it, you kind of see this corona, and it looks a little bit like a flower right there, actually. So that's the endoplasmic reticulum becoming all distended. So it's lost that labyrinth phenoform and become this like distended kind of nebulous outstretching of that nuclear membrane around the nucleus proper. The other thing you're often going to start to see, so we saw actually an intracellular accumulation of quite frankly, water. So there are other types of accumulations than just water. Most of the time, these result from metabolic dysfunction, so lack of enzymatic activity or proper enzymatic activity. And what ends up happening is you begin to see like high levels or dark spots, these accumulations of different types of materials or cellular components that the cells can't either process and metabolize and send out the waste or use for energy or whatever it normally does. And essentially even the backup team, the lysosomes, if you remember, the lysosomes themselves were little vacuoles with tons of pH modifiers and enzymes and whatnot, and they were able to chew up pretty much anything, right? Like a little floating stomach of the cell. So they've usually either become functionally disrupted in one way or another. And there's three categories, general categories of substances that we would see for this intracellular accumulation. The first are what we kind of think of as like these normal body cellular substances. Things like fats and proteins and sugars, some pigments like melanin. You can always see in the case of lysed blood cells, so bilirubin accumulation. So when that's just present in an abnormally high amount, just much larger than usual. We can even see a darkening often in these cell types. So then we have what we consider to be abnormal endogenous projects. Sorry. Uh, so these abnormal exogenous products, so sorry, I'm still getting over this cold. So what these are oftentimes errors of metabolism, like I talked about, and these can come from two different sources. One, these can be based in um, essentially a congenital condition, right? So it can be a gene that has mutations, a gene that's being improperly regulated, proteins or enzymes that are being improperly manufactured or modified. So it's kind of falls into those categories. And then finally we have the exogenous sorry, products. And these are environmental agents. Generally, these are things that can't be broken down and they're introduced. So some can cause problems, right? Too much accumulation of these actually disrupts the normal cellular day-to-day function. However, some of them are seemingly harmless or at least known to be non-toxic. Other thing about some of these accumulations is some of these actually they just need time before they can break down. However, others can stay permanently. So and we'll take a look at some of those examples too. So this is just a little graphic I thought was useful for intracellular uh, accumulations. So as you can see here on this side right here, this cell is undergoing either phagocytosis or pinocytosis, right? It's bringing in endocytosing elements from the environment, and this could be nutrients. This could be some type of extra pigment. It could be endocytosing normal food, and then some of these additional spots. Perhaps there's some type of non-necessary accumulation that's also getting brought in at the same time. But whatever it is that's bringing in, essentially results in cell type number 2 here having these vacuoles essentially that are full of indigestible or unprocessable cellular accumulations. And so that's how we get a cell that's essentially really just clogged up with too much of these materials. And sometimes it's just so much that it's literally, maybe even the mechanism for processing these particular accumulations comes back, right? So it comes back online, perhaps the enzyme gets reactivated or whatnot. But sometimes just because there's so many of these accumulations, they're just clogging up the system, making it hard to process. So we've looked at some accumulations of that were present in cells, right? And I showed you a little graphic representation. And so just kind of the takeaway message is this. These are substances that are either produced or pulled in, endocytosed to a cell, and its processing or whatnot, or removal, right, as waste, exceeds that of its ability to be taken in, and then we just have too much and the system's a little backlogged. So one of the examples they have, like a nice photograph, is one that you may have already had some brief familiarity with from AMP2. So we're looking here at a fatty liver, and what we're looking is we're going to look at a bunch of large they look like blank little circles, and these blank little circles on the inside of these cells are giant, vacuoles just full of fatty acids. And so some of these, and normally your liver does actually have access to fatty acids, and its turnover is important in the role of the liver functioning normally in the body. However, whenever we have abnormal concentrations of fatty acids in the liver, we're able to see in cross sections that first off, the organ becomes larger. Uh, and secondarily, we're able to actually see the cross section. It looks like little bubbles, and I'll show you in the next slide. And so the accumulation of fatty acids in the liver, right, the storing and the sequestering of all of these little fatty acids in the liver can be caused by a number of different metabolic and nutritional disorders. So some including starvation, others are related to alcoholism, right, or metabolic disruption. Here's an example of a fatty liver. So you can see here, let's see if I can grab a good, okay, let's do a red one. You can see here some of these like liver cells over here, generally pretty normal size, pretty normal function. But then you see over here that some of them have just these massive accumulations of white fat droplets, essentially, right? And so we have a normal size liver, and this results in a larger, a too large or a fatty liver. So this is definitely a sign of poor metabolism or nutrition. Um, so accumulations due to genetic disorders of metabolism can sometimes center around normal enzymes that get replaced with a mutant one or a mutated one or one that has just an abnormal function. Um, when that happens, that means the enzyme can no longer maybe break down whichever substance it's normally supposed to be metabolizing, and that substance can not complete all the way through to eventually be eliminated by the cell and begins to accumulate. Um, this also happens in the case of enzymes that are no longer available, perhaps missing or otherwise defunct, right? So these, what we consider to be intermediates or substances which they were supposed to complete their catalysis on, now start to accumulate in the cell. So there are genetic disorders that affect things like, say, glycogen metabolism, which allows to lead to accumulation of glycogen in the cell, or also to the conversion of fatty acids to glycogen in the cell. So a couple of common examples are von Gierke's disease. So this is a type of disorder where glycogen in actually pretty large amounts begin to accumulate in the kidneys and the liver. So what happens is because they lack glucose 6-phosphate, glycogen can't be broken down into true glucose or sugar. And so what happens is the body actually reduces overall blood sugar levels. And the other example it's more topical for sure is that of Tay-Sachs. So this is a genetically linked disorder, and so what happens in this case is there's a malfunction of the ability of cells to turn over lipids, resulting in accumulation of abnormal lipid amounts in the brain or other tissues, and this happens really rapidly, and this causes overall deterioration both mentally, like cognitively, or motor, and results in death usually very, sadly early on between the ages of 2 to 3. Pigment accumulation is generally endogenous pigments, so these are pigments that are within the cell. Some that we can look at right away is icterus, so that's also known as jaundice, just the yellowing. So what happens is it's essentially a bile pigment, but also it leads to kind of a yellow color in the bloodstream. So this is also when the result of when red blood cells are broken down, the heme group actually, bilirubin is a catabolic byproduct thereof, and it results in kind of a general skin tone of being a little bit lighter yellow. Another example is lipofuscin. So it's hard. So some of you already actually know about this one. So this is one that increases on the dermis with age, actually everywhere, but generally you start noticing them on the dermis. Um, so these are kind of this brownish, yellowish brownish pigment, and it's a collection of indigestible cell components that are produced during the normal turnover of cells. Um, and this would be the liver spots that folks begin to see on their skin. Um, some other exogenous pigments, one of the most common ones is that of carbon, simply coming from things like coal dust, often very heavily polluted environments, so coal mines, um, so blackens the lung, leads to tissue damage in the lung. Also poisoning from lead, so blue lead line um, margins start to appear along the gum, so right underneath the teeth. Also tattoos, a very, much more common type of pigment accumulation. So these are pigments that are not soluble, right? And so once they are introduced into the dermis, they're essentially engulfed by macrophages and often become entangled in scar tissue, and they tend to persist for a lifetime. And so that's one of the reasons why if um, the fresh ink that people will get and it looks so nice and sharp and all the lines are really strong, and then you see tattoos that people have had for like 40 years or more, and they start to kind of get really fuzzy and really blurry, and that just has to do with the natural migration of the skin and dermis over time. And so lastly, I just kind of want to show you a picture of this. This is technically pigment accumulation, and this is in the form of silica dust. Um, as you can see, it's kind of preferential, so this would be the entry point for the silica dust, so being breathed in. So it's coming in through the trachea, and it's landing specifically and accumulating, kind of right off the bat, proximally to the intake corridors of the trachea. And it's not only that these are, uh, they're not just colored, right? They're just not pigmented. They're also physically clogged, right? They shut down, they suffocate, they starve, they die. It's just that it's also falling in the pigmentation category because, well, basically there's a color change, right? And so you can see out here in the periphery, there's a kind of newly dying areas of the lung, right? Okay, so that's it for this first set of slides. This continues out through a few more series. And if you have any questions, please don't hesitate to ask.